Folks, time to get started. Once again, Alliston Reid has been engaged in behavioral research since his days at Wofford College. He got his undergraduate degree there, went on to work with John Staden at Duke, got a PhD there while studying psychology. He also studied engineering and computer sciences on his way to a PhD in psychology. He originally then went to Mexico and taught at the Universidad Nacional Autónoma de Mexico, plus or minus, where he taught in Spanish and was, despite the fact that that's not his native tongue, was awarded a number of teaching honors for his teaching in Mexico. He returned from Mexico to the most intellectually stimulating state in the United States, Oregon, in 1985, where he taught at Eastern Oregon University until 1996, when he returned to his undergraduate institution, Wofford, where he is now. He's been chair there since 1998. Alliston has received numerous awards for his teaching. His research has included studies of schedule-induced behaviors, molar optimality theory, neural network modeling, spatial memory, response sequences. I've been delighted to have Alliston as my colleague for, probably at least 20 years. He does what perhaps is most important from a colleague. He continually engages me and stimulates me and indeed gives me a hard time. So I'm happy to introduce him to you, Alliston Reid.
Thank you. Isn't it nice to have a friend like Alan? It is really, really nice to have the opportunity to come here and talk to you about my work. I'm going to be outlining an area of research that I am really passionate about. I love thinking about response sequences, talking about response sequences. So if you like what I'm going to be talking about, please come talk to me. Response sequences have traditionally been explained by a verbal model called chaining theory. Researchers have discussed the limitations of chaining theory for at least 50 years, so I'm not going to discuss those problems now. But one good thing about chaining theory is that it showed how various processes might combine to create structure in behavior. Processes such as conditioned reinforcement, discriminative stimuli, the delay of reinforcement, gradient, perhaps. One of the biggest limitations of chaining theory, however, is that it makes no quantitative predictions at all. No quantitative predictions. So suppose one wanted to create a quantitative model of the way in which response sequences are learned. What would we need to know? What types of experiments can help us? A replacement needs to include quantitative models of effects of stimuli and strength or value of a discriminated operant. When I'm talking about the effects of stimuli, I'm going to be talking about, of course, discriminative stimuli and conditioned reinforcement. When we talk about discriminative stimuli, we have to look in a historical context and realize that when chaining theory was developed long ago, they may have had an idea of what discriminative stimuli were, the concept, but they didn't know that there were different types. They didn't know differences between occasion setters and markers and bridges. So nobody has ever done the research to try to figure out what roles these different stimuli have. In conditioned reinforcement, we know a lot about conditioned reinforcement. In a sense, there are some very good models of conditioned reinforcement out there. I've mentioned three of them here: delay reduction theory, hyperbolic value added model, contextual choice model. But these models have been developed to explain choice and concurrent chain schedules. So one of the questions we will have is to see whether we can borrow some of that some of those models, certain characteristics of those models, and apply them to simple response sequences. With strength or value, we're particularly interested in being able to measure and predict the response strength of individual responses. One of the main questions I'll be having is, can behavioral momentum theory help us here? Now, behavioral momentum theory, as most of you know, is a theory of response strength, and it may allow us to separate some of the Pavlovian versus the operant factors that contribute to response strength or response rate. Then we have the response unit problem. This is the behavioral unit problem. The issue is, how does an entire sequence obtain response strength? If you practice a sequence over and over and over again, sometimes it seems to act like it's like a new behavioral unit itself. So I'm going to be asking the question, what happens when contingencies change? Now, I'm going to be talking about these 4 areas, and I'm going to be going over a good number of experiments somewhat superficially. I'm trying to introduce the topic to you, and I'm trying to show you how you can go do experiments in these areas to be able to identify, to be able to solve some of these questions. I'd like to start by describing a study that helps us understand how discriminative stimuli work in sequences. Here we have well, in this particular experiment, I'm going to be manipulating the presence or absence of a tone in a very simple 3-response sequence. So I alternated 2 types of trials, a trial that had no tone and a trial that had a tone in this first position between the first response and the second response. And I'm going to be asking the question, what does the tone do? Does the tone help improve accuracy at the second position? So the very first response turns the tone on, and the second response turns it off. It makes no difference whether those responses were correct or not. Later on, I moved the tone to the second position to try to find out, well, what would happen if we sort of pull the rug out from under the animal. So let's see what we would expect, what sort of effect we would expect in each different type of sequence. So here are 6 different types of sequences. I didn't use the left, 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 and the right, right, right. The effects of the second response here, we would, if the effects in the second response simply tells us if the tone improves accuracy. And the effects of the third response here tell us or give us some sort of indication of how the tone has such an effect. And to cut to the chase here, the observed effects were exactly what you would expect here, exactly what we expected. In every case, we observed changes in accuracy that were consistent with the hypothesis that the tone tells the animal to press a particular lever rather than to switch to the other side or than to persist. So the tone did improve the accuracy of the second response. And an additional finding that surprised me was that this discriminative stimulus influenced more than just the next response in the sequence. It also improved the accuracy of the third response, even beyond that expected by the improved accuracy to the second response. A little bit more formally, the probability of getting the third response correct, given that the second response was correct in the presence of the tone, was greater than that same probability without the tone. Let's now turn to the issue of how stimulus value is learned in these sequences. Here's the traditional idea of a delay of reinforcement gradient. So you have 3 different responses separated in time a little bit, just a single response here, another response, and we have strength or value. When I talk about strength, I'm referring to the idea of the reinforcing strength of reinforcement on a particular response at a particular position. And by value here, I'm referring to the value of the stimuli associated with that response. And when I talk about value, of course, we're talking about conditioned reinforcement. Now, I've had a lot of trouble applying existing models of condition reinforcement to response sequences because these models have been developed to explain choice and concurrent chain schedules. So let's look at delay reduction theory as an example. Delay reduction theory includes the matching equation as part of the model, and matching within a sequence here, within a response sequence, is not so useful to us. So let's just consider the delay, just the kernel, the DRT kernel, which is the little equation here. I won't go over this in detail. If we assume that responses occur about once per second here, and it takes us a couple of seconds for the animal to eat the pellet and then return to start responding once again, the DRT kernel actually predicts a linear delay of reinforcement gradient, which is not consistent with most people's expectations. Now, it would certainly be nice to be able to measure this gradient, and some of you may have developed some ways of doing that. So let me describe an experiment now that helps us understand a little bit more about stimulus value in these response sequences. I'm going to be talking about an experiment in which I train pigeons to peck red, green, white in that order, let's say. Different pigeons have different orders. And I have imposed 3-second delays between the responses. So 3 key box, 1 key comes on. This is a successive stimulus array. So each time, well, a light will come on. It'll be a red light. Pigeon pecks that. The light goes off for 3 seconds. Another light will come on. It'll be green. Just pecks that. Goes off. Another light comes on. It's white, followed directly by food. So it's a very simple thing for a pigeon to complete. It's sort of like just peck the bouncing ball. I am interested in measuring the difference here in value between red and white. So I've imposed these 3-second delays here to try to increase the difference in value between these. Now, conditioned reinforcement is assumed to be due to stimulus reinforcement relations. So notice that 2 different Pavlovian conditions exist here, 2 different Pavlovian procedures. We have trace conditioning here for the first stimulus, trace conditioning here for the second stimulus, and short delay conditioning here. And the general notion that most people have, of course, is that we have a delay of reinforcement gradient that's somehow a continuous function. But it would be interesting to know whether you really do get a continuous function like this based if it's true that these Pavlovian procedures are as different as they are in the classical conditioning literature. Okay? So which would have higher value, red or white? In this case, the initial response or the terminal response. I think that most of us would predict that white, the terminal response, would have higher value. So we need to find a way of measuring this value. But let's compare this situation to that of a simultaneous stimulus array. So a simultaneous stimulus array, sometimes called a simultaneous chain. In this case, what we've done is we have maintained the exact same response contingency. Animals still have to peck red in this case, that's the S+. Have to peck green here, it's the S+, and then white. But each time, all 3 stimuli are present, so the animal has to figure out, has to learn something about the sequence order. So this is similar or identical to some of Herb Terrace's procedures. He calls the simultaneous chain. Can you predict what the difference in value, well, in fact, not the difference in value, but which is more valuable to the animal, red or white, the initial link, the initial response, or the terminal response here of the sequence? If you believe that Pavlovian conditioning is responsible for this, we notice that the Pavlovian contingencies are really quite different here. Food is delivered in the presence of all 3 stimuli. Each peck is in the presence of all 3 stimuli. So what we've done is we've done a study in order to try to measure the difference between these, the measure the difference in strength or value of the initial response versus the terminal response in both of these situations. This is the procedure that we used. This is taken straight from behavioral momentum theory, where we have a multiple schedule with different components of the multiple schedule. One of the components of the multiple schedule is the sequence component. The sequence component is exactly what I've just described. We gave the animals lots and lots of experience before we shifted to this, simply completing that red, green, white sequence. So red, green, white food, red, green, white food. And the purpose of the sequence component here, and a duplicate over here, is simply to maintain the difference in value between the initial response and the terminal response of the sequence, if there is a difference in the value. Then we have a preference component here. The preference component is a simple VI 60-second schedule. One schedule, it's not two. So we're going to be having a red light come on and a white light simultaneously, and both of them act like the exact same response for the contingency. Peck to white is paid off from the exact same VI 60-second schedule as a peck to red. The only reason whatsoever that the animal should prefer one over the other in this preference component would be because of some sort of contribution of the sequence component. The data surprised me a great deal. Here's the successive array. After we measured the preference, we gave free food during the blackout. I'll show you the blackout. I didn't mention that. Here are the little blackout areas. So we give free food in a VT 20-second schedule and a VT 8-second schedule, different times, in order to, as disruptors, and we'd like to find out which has more resistance to change, the initial response or the terminal response. Of course, resistance to change and preference are normally correlated with response strength. And we found, in very clear evidence here, that the initial response was more resistant to change than the terminal response. One animal showed a reversal, but all the others showed the exact same effect, both for the VT20 and the VT8. The initial response was more resistant to change than the terminal response in this successive stimulus array. So then, what do we get in the simultaneous array? We got the exact same thing. The exact same thing. The initial response was more resistant to change than the terminal response. Same bird here, pigeon 46, had the same reversal. Pigeon 11, right, is still running in this experiment, so I don't have the data yet. But this is the exact opposite of what we had expected. We expected, well, greater preference and resistance to change are supposed to be correlated with higher response strength, which we expected to be the terminal response, not the initial response. So these results are not consistent with the delay of reinforcement gradients that I've described, either any of those delay reduction, any of those delay functions. At this point, it's hard to interpret these data. At this point during the talk, it's hard to interpret these data, but they will make a little bit more sense after we look at a couple more studies. And these studies ask the question, what happens to learned response sequences or learned response structures in general when contingencies change? This introduces us to the problem of the behavioral unit once again. If an animal has completed a sequence over and over and over again so much that it has formed a new behavioral unit, does that behavioral unit maintain its integrity when the contingency changes? Well, if the contingencies reinforce the same behavioral pattern that it had before, it's very likely that they would sort of maintain its integrity. We're going to be asking the question, are response level processes responsible for that? Or is it something, a characteristic of a sequence level process, so it's a behavioral unit issue? But if the sequence if the contingency reinforces a behavioral pattern that's not consistent with that behavioral unit, or whatever the sequence was? What if it does require a different pattern? Which processes here allow behavior to adapt? Are they sequence-level processes, or are they response-level processes? My first point here is going to be that response-level processes often masquerade as sequence-level processes. Now, there are going to be a lot of examples of this I could describe, but I want to describe a fantastic set of experiments by Alan Neuringer and his colleagues. This is one of the most exciting papers that I have read. They trained rats to complete 3 response sequences, as in sequences of 3 responses on 3 operanda. We're going to find out that this number 3, the fact that there are 3 different operanda, is important. They reinforced sequence variability in some of the studies. They reinforced stereotypy in some, and they reinforced just any sequence that the animal might generate in some. Following that, they switched to extinction, and the question to the head, and the question that I want to talk about now, is were the sequences similar or different than before? Now, they had lots of other data that are really worth discussing here, but I don't have the time for that. But just remember that they are showing how extinction works, and it's extinction of these response structures. The main result that I want to discuss is that the relative frequency of each sequence was strikingly similar to its level under reinforcement conditions. So here's an example from one of their experiments. It's an experiment 2, in which they had 2 different reinforcement conditions. They reinforced variability with one group of animals, and they had a yoke group that was receiving the same distribution of reinforcement, but without the same variability criterion. The sequence produced that's listed down here on the x-axis is ordered in terms of the relative frequency or the probability that they actually observed. So under the reinforcement conditions, you can see that they are declining here. And one of the fantastic issue here that they demonstrated is that once they switched to extinction, even though the absolute rate of generating these sequences plummeted during extinction, the sequences that were actually generated, given that a sequence was produced, was about the same. It's almost the same here. So the question for us here is, does the stability of these sequence probabilities mean that sequence-level processes are at work? Now, Alan was kind enough to send me some of his data, so I took it and created a decision tree, which is pretty complicated. It has 39 different probabilities in it, but you can see how it's a decision tree, beginning with the first response of the sequence and come down to generate the last response. And I was searching through this to try to find some sort of regularities, and as I did it, I found that the left branch and the right branch were equivalent to one another. So there's a great deal of symmetry in the tree. There's more than that, because redundancy as well. If we look at the red circles here, each one of these subtrees is almost identical to one another. And we have another group of essentially identical subtrees, identified in blue, and the very first response that was made is unique. Well, so the entire tree was composed of only 3 different subtrees. The first choice reflected beginning of trial cues or end of trial cues from the last trial. The left and right presses were always followed by a red subtree. Key presses were always followed by a blue subtree. Turned out that the transition probabilities depended only on the selection of the prior response. That's very useful information for us because it demonstrates that for this particular schedule of reinforcement, a first-order model should adequately describe choice. And from our point of view here, from my point of view, it's really important to point out that all of these are response-level processes, not sequence-level processes. So here's a simple model that allows us to combine each of these processes. You've seen models like this before. This is It's like a Markov model. I'm calling it a pseudo-Markov model. And inside this, I've placed all these different transition probabilities, conditional probabilities of transition from one response to the next here that reflected the values that were actually obtained here. And you have different cues, stimulus cues, representing stimulus control, that indicate sort of an unconditional probability for the beginning of the trial. So the symmetry and the redundancy in this tree here, the large tree, since it only had subtrees, 3 subtrees, allows us to reduce this to 3 response level rules represented by 4 parameters. The first rule is the rats should be approximately indifferent between left and right, which means that the conditional transition probabilities here involved in left and right should be the same. Like avoid the key after left or right. So we have a low transition probability here for going to the key. And after the key response, prefer to persist on the key than to switch to left or right. And of course, that requires 2 different transition probabilities. The persist on the same key versus switch to the left or right from the key. Now, these are just 4 probabilities now, instead of the 39 probabilities that we had before. Now, a post hoc model with these values, if we're correct here, should be sufficient to reproduce the hierarchy that was used to generate the model. And a close fit here, which you can see we have a relatively close fit, some deviations. A close fit here demonstrates that the 3 response level processes that we've just identified are sufficient descriptions of the data. But that's not too surprising because we use these data to generate this particular model. We want to know how extinction affects response sequences. Are they equivalent to changing different values of these same 4 parameters, or do we need additional parameters? And the answer was that simply tweaking 1 parameter is sufficient. Tweaking 1 parameter is sufficient. We don't need to add any additional complexity to generate the extinction data. Even the sort of weird points like key response are reproduced. The parameter that we tweaked here is the exact same parameter that Nurger and his colleagues identified, and that is that extinction increases the probability of very low, increases the frequency of very low probability responses, which was the key press. So what have we learned here? My second point here is that sequence level stability may be produced by response level processes. Sequence level processes may not be involved. Next point is that stability of the sequences across conditions here is not sufficient to demonstrate that they are behavioral units. But of course, stability across conditions has often been the primary justification for the claim that behavioral units have formed. A little over 10 years ago, 12 years ago, when I was becoming interested in response sequences, I asked a group of researchers what should have been a very simple question. Which would be easier to learn if you train animals to complete a 3-response sequence on 2 levers this time, and then after they've learned it really well, we switch to a new target sequence that differs from the training sequence in only one way, which is easier to learn. This new target sequence requires a different response only in the first position, versus this sequence requires a different response only in the 3rd position. The other 2 responses are identical. We don't give any feedback at all while the animals are completing a sequence, so they learn something only at the end of a sequence. They get every sequence, every 3rd response will end with food or timeout. So which would be easier to learn? Well, I found out, to my surprise, that about half of the researchers at this conference said this would be easier to learn, and about half of them said the other would be easier to learn. and they all had the same justification, which was chaining theory. It turns out that chaining theory, that both answers are perfectly consistent with chaining theory. As a verbal model, chaining does not have the quantitative values of the delay gradient or the quantitative values of the effects of non-reinforcement necessary for us to be able to predict which one of these two would be easier to learn. Now, once I published these data back in '94, everybody saw them and said, oh, I knew that. Right? So which is easier to learn? This is a complicated figure, but it looks much more complicated than it really is. This is what's going on in the first response position of the sequence, the middle response position, and the last response position. No changes ever occurred in the middle response position here. But if we look at the last response position, that's where most of the work is occurring, we notice that some of the new sequences required the same response in that last position, represented by this top line, and some of the sequences in the new target, sequences required a different response. So you would expect it to change. So this is a persistence measure. This is simply persistence. How long did they persist in the same response in that particular position? And we see here that there was not much persistence here in the last response position, the one most contiguous with food. But in the first response position, it took them a long time to learn that the contingency had changed, in a sense, that this first response position seemed to be very insensitive to the change in the contingency. Now, you'd think that the last response would be the strongest, but it changes the fastest when the contingency is changed. Here's another study that confirms this effect, or this result. Remember, we're asking the question still, what happens to learned response structures when contingencies change? When I talked about Alan Nuringer's data here, we're talking about switches to extinction, and now we're talking about, once again, switches to a contingency that no longer reinforces the same one, but, let's see here. So we're asking the question, in this case, well, go over the procedure in a little bit of detail. This is very similar to what we have just done. We trained rats to complete a 3-response sequence on 2 levers. I've got 2 levers in red on purpose until accuracy is high and stable. And then we switch without warning to a new target sequence selected at random, very similar to what we had done before, but in this case we're switching every day. Before, we trained the animals for a long time and then switched one time and let them learn that sequence. This time, every single day, after they've learned a particular sequence like left, right, left, they've had a couple of months of training, left, right, left, then every single day, in the middle of the session, we'll switch to a new target sequence. So that gives us the opportunity to look at what happens before the switch and what happens after the switch, including during those trials before they've even been reinforced. The question, of course, is the same we had before. Are the response positions in the sequence equally sensitive to the change in contingency? And the answer is a very strong no. The top panel here shows the same persistence measure that I showed before. It's just that I have all the curves in the same graph. And as you can see here, the third response here, the response occurring in the third position, did not persist nearly as long as the first response position did. Each time the new training sequence, the new target sequence required the same response. Well, they generally got the same response. They persisted, but when it required a different response, they did change. And all of this is within, say, 100 trials of the same session each time. So the difference in height between these little open circles here for the different response in the third position and the filled circles here, where it's requiring the same response, that difference in height here is sort of a sensitivity measure. It shows how rapidly they adapted to the contingency. And that's what I have here. I've just subtracted these two, so now I have a sensitivity measure. And we can see that the third response position here was much more sensitive to the change in contingency than the first or the second position. So this is consistent with, say, a backward progression of extinction within the sequence. Now this is a dual effect, in this case, of reinforcement and extinction, because they're no longer being reinforced for the prior sequence, but they may be receiving reinforcement as they're learning a new sequence. We can see how extinction alone affects these responses, each response position, by looking how long responding persisted during extinction for each one of these. So for each session here, we counted the number of trials until that subject changed the response in the first or the second or the third position. In fact, we counted the number of trials for the first time they made a change, and we counted for the fourth time they made a change, and the sixth and the 10th. And we ignored this does not include any reinforced trial, so this is just an effect of extinction in this case. So as you can see, the very first response position here was much more persistent than the second or third response positions. I'm not sure why there was no difference between those two. I actually expected to see a bigger difference with the third response position, but this is once again consistent with the backward progression of extinction. Now we wanted to know what processes were responsible for errors that were produced during training in this experiment. And the animals were responding at 80% correct, 90% correct. However, they did generate errors sometimes. They produced the wrong sequence. So what processes were responsible for that? Were they response level processes or sequence level processes? And Catania, in 1971, gave us an idea of where to look. Catania's hypothesis was that reinforcement strengthens each operant independently. When you have a sequence or a pattern of behavior followed by reinforcement, then reinforcement strengthens each one of those instances, each one of those operants separately. Therefore, if you have two response levers only, two levers, but a three response sequence, you're going to have some duplicates of those responses. So requiring two left lever presses should make left more probable in later sequences because of this strengthening effect of reinforcement. I have these represented as ABA and ABB and so forth, but to try to simplify this, ABA represents left, right, left, but it also represents right, left, right. The idea is that this is a sequence that begins and ends with the same response. Here's another sequence that begins with one response and has two other responses. Those are the duplicates. Now, it turns out that errors in sequence production strongly favored sequences that shared the same duplicated response, exactly as Charlie Catania argued years ago. You can see that by the same being much higher than the different. So this finding confirms Catania's hypothesis. It helps us understand which sequences would be produced when errors are made. Well, another source of errors is when the animal fails to notice the end of the sequence, the end of trial cues. So we studied this by systematically degrading the end of trial cues and looking to see what sequences occurred following this. So here's another very simple experiment. In this case, once again, it's three response sequence required on two different levers, and we trained the animals in a situation of a normal trial in which food and a brief tone, were always presented together. After they've learned this very well, then I partially degrade it or severely degrade the end of trial cues. The way we did that was to have the tone paired with non-reinforcement, the absence of reinforcement, or no tone and no reinforcement. Ask the question, how does degrading the end of trial cues influence which sequence was generated next? Now these cues, these end of trial cues, had strong effects. I'm not going to spend much time talking about these particular effects because I want to identify an effect that we found even stronger than that on this particular study. The effect was the training sequence itself. If the animals were trained on ABA, it produced a distribution of errors on the other types of sequences. So you can see for each type of condition that we had, how many errors occurred at this type, and so forth. Notice how different these different distributions were, how the errors depended upon the training sequence. That effect is much greater than the effect of simply the end of trial cues. You can see how the end of trial cues here has an effect there, but that's not nearly enough to account for the major differences in these different distributions. So what causes all these different error distributions to be different from one another? Well, it turns out to be Catania's hypothesis again. Now this is a very busy picture. You don't have to, all you need to do here is compare the heights of these curves. Notice that in 33 out of 36 conditions, the filled bars are higher than the striped bars. Errors in sequence production strongly favored sequences that shared the same duplicated response. As you can see, this effect was very strong. So this is another example of response level processes determining which sequence would be generated. My conclusions: even though organisms may form complex behavioral units, and we know that they do, it appears that animals adapt to these changing contingencies through a combination of response-level processes rather than through processes that maintain the strength of the complex behavioral unit. The problem with the behavioral unit is not so bad, after all. It does not prevent us from studying response structure. I believe a lot of people have not worked in the areas of response sequences and not asked questions about response structure simply because of this problem with the behavioral unit, that it seemed to be intractable. How can you have a model of reinforcement if the unit on which reinforcement is acting is changing? It's a very difficult problem. Well, it turns out that you can learn a lot about the ways in which response structure changes over time, changes as we change conditions, for example, change contingencies, without having to worry about some sort of response strength for the entire unit being maintained. We need to reconcile our predictions of value or strength based on the delay of reinforcement gradient with the actual data. The initial response, we found that the initial response was more resistant to change than the terminal response in 3 situations here: when the sequence was switched to extinction, and when we switched to a new target sequence, and in preference tests when given extra food. Catania 1971 was right. Reinforcement strengthens each preceding response independently. Requiring 2 left presses in a reinforced sequence causes their strengths to combine, making left presses even more likely in future sequences. Now, for those of you that are looking for a place to do research, this is a great area. The questions are historically important. People have been relying on, been interested in response sequences and have been trying to explain response sequences with chaining theory for 100 years or so, or more. There are lots of unanswered questions, and it's not really clear to me why we still have so many unanswered questions. I've tried to identify some of those questions today, try to give you an idea of how you can go in and start working on those and try to answer the questions. We have the technology, as they say on TV. We have the procedures necessary to be able to figure out what's going on. One of the things that I like so much about this area is that it encourages a broad overview of this area. It allows you to work in condition reinforcement, or at least consider how condition reinforcement contributes to response structure. It allows you to work in the idea of response strength or learned value of a response or learned value of the stimuli here. It allows you to ask really interesting questions about the way in which delays of reinforcement affect behavior. What I'm hoping people will start doing when they're asking questions about response sequences is to try to identify the Pavlovian processes responsible for that. It's a mystery to me why it is that we found the same type of resistance to change functions when we had the simultaneous array and the successive array. According to, if Pavlovian rules, if Pavlovian processes are the ones that are supposed to be responsible, they should not be the same. They should not be the same, yet they are. We have issues with stimulus control. Nobody has asked the questions about how do these discriminative stimuli actually convey information to the animal, or do they just set the occasion for the next response, or do they act as marking stimuli, or act as bridging? Do they do all of these things, or what do they do? And of course we have the assignment of credit problem that's inherent anytime you have patterns of behavior that are reinforced, so where the reinforcement animal has to figure out, well, speaking very informally, the animal has to figure out what produced the reinforcement. Was it a whole pattern of behavior, an individual response, an individual operator, what have you? Now, I teach, and as a professor, I always give a homework assignment. So I want to give you a homework assignment. You've got lots of time to do this. Design and complete at least one experiment that contributes to our understanding of response sequences. Present it here next year. Thank you very much. Easy questions.
Here we go. Charlie's at the top.
Charlie!
Can I go to the microphone? Sure. Is that well, Elliot Shimoff and I would get out of the homework assignment, so it's just too great a temptation to mention one experiment that I hope would contribute to this, and that involves the question of condition reinforcement. We were thinking about the FI as involving a stimulus, which when it comes on is going to be a condition reinforcer, but the onset should be a weaker condition reinforcer if it's a long FI than if it's a short FI. The onset of suppose you have an FI on green, and it's a 10-second FI. The onset of 10-second green light should be a potent condition reinforcer compared to the onset of a green if it's a 60-second FI. So we did some observer response experiments in which there was a short time period when a second key was available, and if there was no pecking on that key, then the scheduled key came on yellow, at the end of which there was either extinction or reinforcement. And there was a certain time value, which was also going to be the value of the FI. If the observer key was pegged, then you get the green coming on if FI was scheduled on that particular trial, and you have red coming on if extinction was scheduled in that trial. And we looked at the rate responding on the presence of this other observing key, and in fact it was the same function as you get for delay reinforcement functions. It was a log function, a linear function on semi-log coordinates. So it does suggest that the condition reinforcing effect, if you're looking at rates that are producing these stimuli, do change as a function of the strength of the condition reinforcer, which is measured by time from the stimulus onset to the reinforcer. So
that's very nice.
So that means I don't have to worry about that here.
We can still do another.
Well, I'm not sure this is resistance to change and behavioral momentum stuff. What you're saying, when you have this, is a fairly short in terms of overall duration sequence, and it's the lead response that seems to be the most resistant to change in a number of different aspects. If you bring that down to the level of a single peck, stand in front of the key, move toward it, that basically says that it's initiating the operator. It's going to be something disruptive. And in a way that makes a lot of sense to me. I'm not sure quite how you follow it up. But you have to get into stuff like further analyses of your sequences. Francis Mechner's work on revealed operands should be interesting here. But there, it doesn't hang together as well because it's the initiation of this. Francis over here, the initiation of this, the one response and then a series of others, and then a sort of exit response. Okay, I've finished this thing. It's the initiation that is the most disruptive. But what you've done is to look at which response rather than whether response in many of these cases. And I'm not sure where this is going to go. Boy, I just love the idea that if you're in a situation where your behavior is being disrupted, what you're going to do is move up towards initiating this particular response. That's going to be the most resistant rather than...
Could you repeat the point, please, so it will get on the tape?
My point...
Oh, no, if he could. Or if you could go to the microphone, or if he could do it.
It's the wrong point.
I just think this is a wonderfully exciting and challenging series of results. And what it says to me is that even though it's not what I would have expected in terms of delay of reinforcement, conditioned reinforcement, stimulus control, and resistance to change, the idea that it's the lead response in a fairly short sequence that turns out to be the most resistant to disruption in the sense of its stimulus is preferred, it is harder to replace with another response, suggests that when we have a well-trained operator, we just look at a single unit, like emitting a peck, which has some temporal properties, you move toward the key, you approach it closer and closer. Getting that started is what's going to be most resistant to change if it's been reinforced frequently. And what I've been trying to fumble with is how to reconcile that with what else I know about initiating chains and time involved, the latency to get started on that unit. And I haven't figured that one out.
Well, one of the characteristics of behavioral units in the past, like the way Barry Schwartz has talked about the behavioral units in his fairly complex sequences, has been that when he would train the animals to complete this sequence, they would learn it very, very well. He would try to define it as a behavioral unit by measuring both stability, like I've been critical of here, but also latency issues, switches them to extinction, and once they start the sequence, they tend to run off the sequence fairly. The latency would not change within the sequence, but it is the first response that was the sort of the weakest in the sense that it was the one that was that showed the least resistance to change. It was the one that changed the most. So Barry Schwartz argued that these are behavioral units partially because the integrity of the sequence, once they start the sequence, tends to run off some little computer program in a sense, but getting initiated, making that very first response was where most of the effect was occurring. So, but of course I have the exact opposite the findings here.
That's
one or two more questions. Maybe sort of a free flow approach. I'm going to suggest over time to the teacher.
Go to the microphone then. I'll accept a quid pro quo only if you really promise to complete this assignment.
I was saying quid pro quo, so homework assignment, harder one, I guess. Can you conceive a sequence level process that is irreducible to a response level process?
Yes. Several people in the audience here have already demonstrated the matching relation of sequences, where a group of you would have a response sequence, and you get sequence level matching, but you would not get matching of the individual responses within that sequence.
Well, the problem is that, as you know, if you use the first order Markov chains, you can go backwards, and all of a sudden, including more of the stimulus control of the next response. The Markov chain, as you mentioned, first order means that the next response is influenced just by the current response. Or, if you want, the current response is influenced by the previous response. Now, nothing precludes the fact that other responses can influence the current response, not just the last one, maybe the last two.
Correct.
And I cannot prove it, but I think it is not possible to have a sequence level process that is irreducible in principle to a response level process if you give sufficient latitude to the degree of stimulus control.
Okay, I agree with that, but I would also argue that if you define a sequence level process as that process where if you're simulating it with a Markov model, if you have to go back in time to include every response of that sequence, then that is a sequence level process. If you're going back only a certain amount of time, but only dealing with some of the constituent responses within that sequence, I would argue then it's still a response level process.
So my last question is this. It seems to me that the sensitivity of the first response in the sequence or the last response tells you more about which stimulus is controlling the response than anything else. So the beginning of the sequence has generally some kind of time marker, or timeout, or tone as salient stimulus, very different from the reinforcer, whatever came before, or something like the intertrial interval. So it's that is the cue for the first response, whereas the last response is preceded by other similar responses. Now, one thing that you need to worry when you report to Schwartz experiments is that if the animal, for example, is engaging less and less in the operant sequence, is preventing the extinction of subsequent responses. Because suppose that I learn to do left, right, right. Now, if the first component is decreasing because in extinction the animal starts to do other things, like turning around, it's not pressing the levers, naturally the left, it's doing other things, and it's preempting the opportunity to extinguish subsequent behavior once it starts, right? Because it's not starting the operant. The links later on down the chain, if you want to use as a descriptive term, the links down the chain would not have the opportunity to extinguish.
I don't actually buy the argument because of Alan Nuringer's data. Alan has demonstrated, well, with the data that I showed, he demonstrated that that the sequences were maintained during extinction, so that the rank ordering of sequence probabilities were maintained, even though extinction reduced the rate of generating those almost down to zero. The conditional probability of producing a particular sequence, given that some sequence was going to be produced, was almost maintained.